Welcome to an introduction to cell division, part 2 of 3 for the Alberta Science 30 curriculum. This covers topics in Unit 2, the biology unit. Mitosis involves 5 stages. Meiosis involves 9 stages. Mitosis is mainly involved in growth, maintenance, and repair of cells and organisms, whereas meiosis is only for gamete formation, so you get sperms and eggs out of it. The result of mitosis is one 2N cell forms two 2N cells. In meiosis, one 2N cell produces 4 N cells. 2 diploid cells are formed for mitosis. So one 2N cell produces 2 2N cells. And 4 haploid cells form in meiosis. One 2N cells produce 4 N cells. Remember that N is the complement of chromosomes. Therefore, for instance, in the case of humans, if you start with 46 chromosomes in mitosis, you end up with 2 cells that have 46 chromosomes each. For meiosis, if you start with a cell that has 46 chromosomes, your end result are 4 cells that have only 23 chromosomes, so half the number of chromosomes as the original cell started with. Mitosis occurs everywhere in the body, whereas meiosis only occurs in gonads, so the ovaries and testes of individuals. Meiosis is the main cause of existing genetic variation in animal and plant populations. Mitosis in the cell cycle. Primarily, most of the time the cell exists in this state called interphase. Interphase is divided up into 3 separate phases. The first one is G1, which is a growth phase, so the cell gets bigger and it replicates things that it's going to need for DNA synthesis. Then, during the S phase, the DNA is synthesized. Here you're copying the DNA and getting ready for cell division. DNA synthesis is necessary so that the chromosome numbers stay the same. If you're talking about mitosis, remember that you need to, for human cells, need to start with 46 chromosomes and end with 46 chromosomes. So you're going to have to double the amount of DNA that you have during the S phase. The third part, or G2, is growth 2. So again, the cell is growing and getting ready for cell division. Mitosis consists of 4 separate stages, PMAT, so Prophase, Metaphase, Anaphase, and Telophase. This mitotic phase can takes about 1/24 or 1 out of 24 hours in a day. When mitosis is complete, there is a phase, or there is a process called cytokinesis where the cells divide. The average cell living 24 hours divides for about 1 hour. Approximately 23 hours is spent doing other things. So, let's just say you have a cell that divides every 24 hours. So 1/24 of that time is spent dividing. 3/24 of that time is spent doing the job that the cells are supposed to be doing. For instance, producing hormones or enzymes for cellular or for food digestion or things like that. During interphase, chromosomes are not visible, so you can't see them. DNA is in its chromatin form. What are the main events? In G1, there is growth and protein synthesis. In S phase, there is DNA replication and sister chromatids form. And in G2, there is growth and protein synthesis. This one says, dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. So, as it's approaching mitosis, the chromosome or the chromatin condenses into chromosomes. In prophase, you start to get something called spindle formation. The contents of the nucleus become visible as chromosomes and nuclear membranes disappear. So that chromatin starts to condense into chromosomes, and the nuclear membrane, so the thing that makes, or the membrane that separates the nucleus from the rest of the cells, disappears or dissipates. Centrioles, or these things that organize cell division, migrate to the opposite ends of the cell, or opposite poles of the cell. And these things called spindle fibers form. These are made out of actin and myosin. These are structural proteins whose job it is to guide the chromosomes to opposite ends of the cell. And the reason why they do this is to make sure that cell division happens in an orderly way and you only get one copy to each side of the cell. Things called asters or more microtubules radiate. That's right, spindle fibers are microtubules. They're not actin and myosin. I'm sorry. These things called asters radiate from the centrioles. Chromosomes are made up at this point of two sister chromatids held together by a centromere. Remember that those sister chromatids will separate and get pulled to separate sides of the cell. There are two stages of prophase. There's an early stage and a late stage. In the early stage, you have this mitotic spindle forming out of microtubules. You have these little soda can shaped things called centrioles forming, and they organize the two halves of the cell that will eventually split. These chromosome pairs consist of sister chromatids, and as it progresses into late prophase, you have the nuclear membrane disappearing. You have these microtubules attaching to these chromosomes, these sister chromatids, and starting to push them towards the center of the cell. In metaphase, metaphase is signified by when these chromosomes line up in the middle of the cell. And the middle of the cell is referred to as the metaphase plate. Replicated chromosomes line up along the equatorial plate, again metaphase plate. Individual chromosomes can be seen since they are lined up, so you can see 1, 2, 3, 4. The number of spindle fibers equal the number of chromosomes in a diploid cell. So, in humans, you would have 46 spindle fibers because you have 46 chromosomes in a diploid cell. So there must be 46 spindle fibers. In anaphase, the centromeres divide, so they start pulling these sister chromatids apart, and daughter chromosomes are pulled to separate poles. Chromatids, now known as chromosomes. So here are your chromosomes. They move to opposite poles. Assuming everything goes well, the same number of single-stranded chromosomes are found at each pole. So you have the same number of chromosomes at this pole as you would at this pole, 4 and 4. So if 2n is equal to 12, the number of chromosomes at each pole should be 12. The chromosomes reach opposite poles in telophase, so when they hit those opposite poles, it's the beginning of telophase, or cell splitting. Chromosomes begin to lengthen out again and become chromatin, so they're starting to get released from their histone organizational protein structure. the nucleus starts to reform, the nuclear envelope starts to form, and you start to get this indentation in the cell, especially in eukaryotic cells, animal cells, called a cleavage furrow. This cleavage furrow isn't seen in plants. The spindle fibers dissolve, the nuclear membrane reappears, cytoplasm divides in a process called cytokinesis. So when one cell divides into two, it is called cytokinesis. In cytokinesis, cytoplasm pinches in or invaginates. That's the word that scientists use in animal cells. A cell plate is formed in plant cells. So you can see right there, you have that really thin line. That is a cell wall and a cell membrane starting to be formed. That's called the cell plate. The cell plate will eventually become the cell wall, which is made up of cellulose in plants. So here you have you have two cells here and here. You can start to see centrioles forming. So that would be the starting of prophase. You can still see the nuclear membrane. So it's early prophase. So none of these pictures really show interphase. Here you have the chromosomes lining up along the middle. That's along the metaphase plate, so that's metaphase. Here you have these are sister chromatids at this point. Here the sister chromatids have separated into chromosomes, and the chromosomes are being pulled to the poles of the cell. So this is anaphase. Here you have a late prophase cell because they're not lined up like they are in metaphase. So they're sort of pushing and pulling and trying to find the center here. Here you have those chromosomes have reached the edge, and you have this invagination and cytokinesis starting to occur. So that's telophase. Here you have a cell starting to enter interphase, where you have the nuclear membrane reforming and these centrioles and spindle fibers starting to get taken apart until the next time these cells have to divide. And then, so here is like a little picture. Here's where you screwed up. Telophase comes after metaphase. So it's something that you'll have to memorize. And here, I'm appalled at what they will put on television nowadays. It's nothing but senseless viruses and gratuitous dividing. So it's sort of like a play on